In this exercise, we're talking about microbial growth. Certain things can be added to optimize the growth of the microorganisms. Some things you have to look at the various parameters and make sure you're within the proper range, like pH. For bacteria, you don't want it too low. They don't like that. They like it around pH 7 to pH 8. But fungi, they prefer more acidic pH. And so you have to look at what are you growing, what are the optimum growth requirements for that particular organism. So pH will be critical. For most organisms, it can grow within a certain range, but they have an optimum within that range for the pH. Same thing with temperature. There's usually a range they can grow up, but there's going to be an optimum temperature. You can often look at where is the organism naturally found. That's going to give you a clue as to what the temperature optimum is going to be. If something is naturally found in the human body, it's probably going to prefer a temperature around 37 degrees C. But if it naturally is found in the Arctic Ocean, it's probably going to prefer something closer to freezing. What are the nutrients? Is it specific the type of nitrogen? What about certainly the carbon source? What about phosphorus and calcium and sodium? So you have to look at all of those various nutrients and oxygen requirements. Some organisms have to have oxygen. Some organisms, oxygen will actually kill them. They're anaerobic, and so you have to remove the oxygen. Some organisms, like Streptococcus, it can grow in oxygen, but it really doesn't like it at the atmospheric level. It's a little too strong for it, so it prefers a reduced level of oxygen. So you have to make those accommodations for it. And then some organisms, they're like, well, if oxygen is there, fine. If not, that's fine too. It can take it either way. So when you are working, certainly with a particular, say, bacteria, you need to know what its optimum growth requirements are. For bacterial growth, bacteria do not go through mitosis. They grow, the majority of them, by binary fission. Some have other means, but the majority of them go through binary fission. Basically, after DNA replication occurs, the cell's going to elongate. The DNA and the copy, they go to opposite ends of the cell. You can have a pinching in of the membrane and cell wall, and then bingo, you've got two new cells. So when we talk about bacterial growth, we're talking about the increase in the number of the viable or living cells. Generation time. This is often sometimes called doubling time. How long does it take for the number to double? So you have your bacterial population, and you have, let's say, 100 cells. How long does it take for you to get to 200 cells? And usually you are looking within a certain portion of the growth curve on this. So the growth phases of the bacteria is divided into 4 different phases. The first one is lag, then there's log, also known as the exponential phase, the stationary phase, and then last is the death or decline phase. So when we look at a graph, it's graphed this way. You have a brand new cell. It starts out down here in the lag phase. It's adjusting to the new environments, brand new cells. Maybe you just put it into some fresh nutrients. It's adjusting to that. And then during the log or exponential phase, you have this rapid increase in the number. It is during the log phase where you are going to be determining the doubling time or the generation time. Typically what happens is you start to run out of nutrients, and that's when the cell enters into the stationary phase. You have basically the same number of cells being produced as those that are dying. They're in equilibrium. Now you reach a certain point, you're definitely running out of nutrients, and you have more cells dying than you do producing new cells. So this is this decline or the death phase. This is a classic growth curve at this point. How long does it take to go through this? It's going to vary depending on, number one, the particular bacteria you're talking about. It's going to depend on the growth conditions. What is the pH? What is the temperature? How many nutrients are there, etc. So how do we determine what the concentration of the bacteria are? How do we determine the number? There are a couple different ways you can do it. Probably the most common way is to take your sample and dilute it. We've already talked about dilution some. Remember, you're going to take a sample and you're going to basically put samples on petri dishes, let it grow, and count the number of colonies. Remember, you only count between 30 and 300. So that usually means you're going to have to dilute your sample. Once you have diluted the sample, you either put the sample on a spread plate or a pour plate, depending on your resources. Do you want to grow it just on the surface? Do a spread plate. If you want it to be able to grow within the media, then you do a pour plate. Another way that you can determine the bacterial concentration is using a spectrophotometer. That is an example of our spectrophotometer is shown here. What a spectrophotometer does is it takes a sample, you put it in this little spot here. It's in a little glass tube, it's called a cuvette, and you put it in here, and then there's a light beam that shines through. The idea basically with the spectrophotometer is that if you have a broth and no bacteria are growing, that broth should be nice and clear, so all the light should be able to penetrate and pass through the sample. And then you measure, this is going to give you a number up here, it will measure essentially the amount of light that's passing through. So most of it should get through if there's nothing growing in it. As you know, bacteria, when they grow in a broth, it turns cloudy or turbid. That cloudiness, when you put the sample in the spectrophotometer here, and the light goes to shine through it, now if there are bacteria growing, making it cloudy, not all the light's going to be able to pass through. So less light passes through, and once again, you're measuring that. The amount of light that's able to pass through, we get these measurements, and we can correlate that to, the first time you do it, you have to also be running dilutions and doing spread plates, and you can relate the number, this reading that you get from the spectrophotometer, that number of, which relates to how much light is passing through, you can then relate that to the number of bacteria in that sample. And so then the advantage of using a spectrophotometer is if you have a lot of samples to measure and you want to know the concentration, once you have figured out this relationship of the number, this reading, this absorbance number, to how many bacteria, you just take your sample, put it in here, bing, get your number, bing, get the next number, just one after another, run your samples through, and then all those numbers that you have collected, you plug it in a formula where you're able to relate it and come up with the bacterial concentration. So if you just have a few samples, you can just do the normal dilution, pour plate, or spread plate. If you have a lot of samples and you have access to a spectrophotometer, that may be the route that you want to go. But it all relates to determining how many bacteria essentially do you have. It's relating to that growth of them. You start with a small number, the growing, the number's increasing. What is that number then? So this are a couple ways that you can use to determine that.